Rate of photosynthesis, required practical B4. Tips for planning a practical. If there are diagrams provided, they're extremely important. Use them, annotate them. The 3 really important things that you should do associated with a method is state the variables, the equipment linked to those variables, and when collecting results, you repeat. You could also be asked about control variables, controls, and safety. The aim of the investigation for photosynthesis is investigate the effect of light intensity on the rate of photosynthesis, and a diagram has been provided. 2 key pieces of information. Firstly, you want to annotate the aim. They give you the 2 variables in the aim: light intensity and the rate of photosynthesis. So we're investigating how light intensity affects the rate of photosynthesis. Then you want to use the diagram, annotate it. Regarding light intensity, well, they've given you a light source. So off the light source, I would write light intensity. For the rate of photosynthesis, you obviously need a plant, and they've provided you with pondweed in sodium hydrogen carbonate solution, and that's inside a boiling tube. So I'd annotate boiling tube. The sodium hydrogen carbonate solution is what's providing carbon dioxide. So again, you can annotate that. The boiling tube and the lamp are sat along a ruler with a scale. And that scale is going up in 10 centimetres. So they've got intervals of 10 centimetres. So again, I would annotate that. What that suggests to me is that then the boiling tube is going to move along that ruler, along those 10 centimetre intervals. By moving the boiling tube, the light intensity from the lamp is going to be reduced. I can already see how this method is going to work just from looking at the diagram and using those 2 key variables. Now, regarding rate of photosynthesis, how do we measure rate of photosynthesis? Well, when plants photosynthesize, they produce oxygen. So what would happen is you'd get oxygen bubbles inside the boiling tube. So I'd actually draw a bubble in the boiling tube. And what we need to be able to do then is you need to be able to count the bubbles in a given set of time. So I'm just going to say count the bubbles within a minute, and that's what I'd annotate. So clearly from here, I've got a gist of what is going on with this investigation. As I stated at the start, you need to be able to state your variables. So your variables are the independent variable, the one that you change, and the dependent variable, the one that you measure. So looking again at that aim, the effect of light intensity on the rate of photosynthesis, the one that I am changing, the one that is the independent variable, would be light intensity, and the one that you measure is rate of photosynthesis. But we need to be able to break it down and link it to this method being provided. So the independent variable, as we said, is light intensity, but how is light intensity changed? Well, it's the distance of the boiling tube from the light source. The rate of photosynthesis for the dependent variable, we said, is the number of bubbles counted per minute. The second thing that you need to now do is state the equipment that's associated with those variables. So with the independent variable, you obviously need a light source, so a lamp or a light source, and for the distance you need a ruler, and I've stated the 10 centimetre intervals. Regarding the dependent variable, the number of bubbles per minute, well, the first part, the number of bubbles, means that we need pondweed in sodium hydrogen carbonate solution inside a boiling tube. And for the minute, it's obviously time, so I need a stop clock. So very simply, I've stated the 2 variables, and then I've stated the equipment that is needed. So what this is telling me then is to investigate the effect of light intensity on the rate of photosynthesis. Pondweed is required in sodium hydrogen carbonate solution inside a boiling tube. You need a light source that is providing the light intensity, and you need a ruler with intervals of 10 centimetres. The boiling tube is going to be moved along that ruler, which will reduce the light intensity, and each distance, the number of bubbles per minute is counted. That is the general step of the method. Give the variables, state the equipment associated with the variables. Now we've introduced this concept of dual coding with the idea of having less words, just images, and using your finger to point to it to tell the story. So very simply, if we look at the diagram, we can easily describe the story by having less words, less annotation. So firstly, next to the lamp is the light source. That's already been given to you. Then you've got the boiling tube. All I've included is a bubble, and I've drawn a stop clock. With the ruler and next to the boiling tube, I've drawn a blue arrow going to the right. So using my finger, I can easily tell the story. So pointing to the light source, I can easily say that the light source is providing the light intensity, and this is the independent variable. How that's achieved is that the boiling tube is then moved along this ruler. Okay, so this is what the arrow's telling me. Boiling tube is moved along, and that distance of each 10 centimetre intervals is the independent variable. Now, the dependent variable is the bubbles that are counted per minute. For this to happen, I need pondweed in sodium hydrocarbonate solution in a boiling tube. So that's one way that you could do the dual coding and keep practicing it. The second way is that you could have the diagram. I'd always draw a bubble inside of it if it's not provided, just to remind you that's what you are counting. And you could easily write the word independent variable, and I've pointed it to the ruler part, because that's the distance being moved along, and the dependent variable I've pointed to the bubble. And then again, I can draw the associated diagrams with it. So here I could say the independent variable is the light intensity, which is the distance the boiling tube is moved along the ruler. And the dependent variable is the number of bubbles produced in a minute. The way that we learn and revise is very key for our understanding. So we now know the two variables. The independent variable is the light intensity, the distance of the boiling tube moved. And the rate of photosynthesis is the number of bubbles counted per minute. What you always need to state in a method, not just for this one, but any method, you need to talk about how to collect the results. And when you're collecting results, you need to say that you're going to repeat it and calculate a mean, usually. So in this scenario, 10 centimetres is the first distance. You would then repeat that 3 times and calculate the mean number of bubbles per minute. You then need to say you're going to repeat it for each distance of the intervals of 10 centimetres. So you're going to repeat it for 20 centimetres, 30 centimetres, 40 centimetres, 50 centimetres. So you always need to repeat for what your given independent variable is. So within your method, you're stating the variables, the equipment associated with it, and the collection of results with the repeating. Now, what they can ask you is they could ask you for some control variables. What you've learned is that photosynthesis is affected by the chlorophyll concentration, it's affected by carbon dioxide concentration and temperature. So therefore, in this investigation, you need to keep temperature controlled, keep it the same. You need to keep the carbon dioxide concentration the same, and you do that through the concentration of the sodium hydrogen carbonate solution. The chlorophyll, you need to make sure that you're using the same pondweed sample each time. Then the last control variable is when you start the investigation for the 10, 20, 30, 40, 50 centimetres, you now need to allow the pondweed to acclimatise to the environment, to the conditions. It needs to be able to start to photosynthesize. So you need to give it time. So it could be a 5-minute time before you do the experiment. Another thing that they can ask you is a control. What we need to ensure is that the method and the equipment that we're using is correct. It's giving us valid results. And to do this, we need to make sure that photosynthesis actually happens because of the presence of light, which we know is true. So control would be to set up this equipment, the pondweed in the sodium hydrogen carbonate solution in a boiling tube with no light source, And what you'll notice is there will be no bubbles. Photosynthesis does not occur. That then tells us that this equipment, this method does work. And the results that we're getting is because of the change in light intensity, and therefore valid results. Another factor they could ask you about within this planning section is safety. And we all know to be safe, we should wear safety goggles. With this practical, the boiling tube is being moved and is obviously made out of glass. So you could use a retort stand, a clamp stand. Thirdly, you're using a lamp, which obviously has electricity running through the cables. So you need to make sure that you are touching the lamp with dry hands. That is us then covered the whole section of planning. What they can actually then do is they could ask you or provide you with a different take of this method, because it would give you valid results. The idea of counting bubbles is quite difficult because there could be a miscount. So instead of counting bubbles, you could measure the length of the bubble using a syringe and a capillary tube, which is seen in this diagram here. All you would need to do here is again annotate it. The independent variable is still going to be light intensity and the distance of this boiling tube moving along your ruler. The dependent variable is still rate of photosynthesis, but you're looking at the length of the bubble in a given time. You need to be able to state your equipment. So again, light intensity and the effect of it, you need a light source, you need a ruler. You then need, for rate of photosynthesis, you need the pond weight in the sodium hydrogen carbonate solution inside your boiling tube. But now you need the capillary and the syringe. And again, you need a ruler here to be able to measure the length of the bubble. Now, they could also look at different ways of measuring rate of photosynthesis, looking at the effect of temperature and carbon dioxide concentration. So with temperature, you could have different boiling tubes placed in a water bath of different temperatures, and again, you could count the number of bubbles. With carbon dioxide concentration, you would then be looking at that sodium hydrogen carbonate solution and having different concentrations of that. And again, then you can then do your measuring. What you would need to be able to do for this is, again, just apply those simple rules. What are your variables? What would be the equipment for those variables? How do you collect the results? And then anything else that they're required of you. So the temperature, obviously, and the carbon dioxide concentration, they are now your independent variables. Your dependent is the measuring of the rate of photosynthesis. How is that done? Is it the counting of number of bubbles? Is it the length of the bubble? So clearly, what you need to know for any planning of a method is you need to know the variables, you need to know the equipment linked to those variables. This then gives you an understanding of the method, and hopefully a diagram is always key to help you to understand that further. You always need to know when you're collecting results that you're repeating for that one and calculating mean, and then you repeat for anything that you're changing within your independent variable. They could ask you about control variables, they can ask you about controls, they can ask you about safety. For this investigation, you need to know the variations on how to improve the practical and the method, but also different factors that can affect the rate of photosynthesis.